Health authorities and international surveillance networks are currently focused on a developing public health event involving a rare and severe respiratory pathogen. On May 2, 2026, the World Health Organization received notification of a cluster of severe acute respiratory illnesses aboard the MV Hondius, a Dutch-flagged cruise ship operating in the South Atlantic. This incident has brought renewed scrutiny to the Andes virus, a specific strain of hantavirus that is increasingly being studied for its unique ability to facilitate human-to-human transmission. Unlike most hantaviruses, which typically require direct contact with rodent excreta, the Andes virus has historically demonstrated the capacity for limited onward spread among humans in close proximity. This characteristic is the primary driver behind current genomic investigations, as scientists work to determine if specific mutations are influencing the severity or the transmission dynamics observed in this latest cluster. The situation began after the vessel departed Ushuaia, Argentina, on April 1, with an itinerary that included remote regions of Antarctica and several Atlantic islands. Public health officials are now tracing the timeline of an outbreak that has already resulted in laboratory-confirmed infections and multiple fatalities. To understand why this development is being treated with such gravity, one must look at the clinical mechanism of hantavirus pulmonary syndrome, or HPS. While the title of some recent reports may use alarmist language, the biological reality is sufficiently serious to warrant intensive monitoring. Hantaviruses are enveloped, negative-sense RNA viruses. When they enter the human body, they do not simply irritate the respiratory tract. They target the endothelial cells that line the blood vessels, particularly within the lungs. This leads to a phenomenon known as vascular leakage. In severe cases, the capillaries in the lungs become so permeable that fluid begins to fill the alveolar spaces, effectively causing the patient to drown internally. This rapid progression from initial flu-like symptoms to full-scale respiratory failure and cardiogenic shock is what characterizes the most dangerous phase of the disease. The case fatality rate for hantavirus pulmonary syndrome in the Americas has historically been estimated as high as 35 to 40%. Making it one of the most lethal zoonotic threats currently under surveillance. The current framing of this event as a genomic concern stems from the ongoing sequencing of the viral samples collected from the MV Hondius passengers. Researchers are examining the virus's L segment, which encodes the RNA-dependent RNA polymerase, to identify any shifts in the viral architecture that might explain the clustering of cases. While some headlines suggest a designed mutation, virologists emphasize that such changes are generally the result of natural evolutionary pressures during spillover events. The primary objective for agencies like the CDC and the European Center for Disease Prevention and Control is to ascertain whether the virus encountered by these travelers possesses any novel traits that enhance its stability in aerosols or its affinity for human cellular receptors. This reporting will now examine the specific developments of the MV Hondius outbreak and the international response triggered by these findings. The progression of the outbreak on the MV Hondius provides a clear window into the logistical and diagnostic challenges posed by hantavirus. According to official reports from the WHO and the CDC, the first suspected case involved an adult male who developed a fever, headache, and gastrointestinal distress on April 6 while the ship was at sea. His condition deteriorated rapidly, and he died on board on April 11. Because no microbiological testing could be performed at that time, his death was not immediately linked to a specific pathogen. It was only after subsequent passengers began displaying similar symptoms, fever, pneumonia, and acute respiratory distress, that international health protocols were activated. By May 2, when the ship was near Cape Verde, a laboratory in South Africa confirmed hantavirus infection in a critically ill passenger who had been medically evacuated. Subsequent genomic sequencing, conducted by May 6, confirmed that the causative agent was indeed the Andes virus. As of mid-May 2026, the total case count has risen to 11 individuals, including 8 laboratory-confirmed cases, 2 probable cases, and 3 deaths. The international response has been swift and multi-layered. The CDC dispatched a specialized team to meet the vessel in the Canary Islands to assess exposure risks among the American passengers on board. Simultaneously, the Spanish and French health ministries have reported their own confirmed cases among returning travelers who were part of the same voyage. A key element of the current investigation is the mapping of the patient's activities in Ushuaia, Argentina, prior to embarkation. The prevailing hypothesis is that the index case likely acquired the infection from an environmental source in South America, where the long-tailed pygmy rice rat serves as the primary reservoir for the Andes virus. However, the timing of symptom onset for subsequent cases, which occurred in clusters, strongly suggests that human-to-human transmission took place within the enclosed environment of the cruise ship. This is a rare occurrence for hantaviruses and is the specific focus of lab technicians currently analyzing the viral load and shedding patterns of the survivors. In terms of laboratory developments, the focus is currently on metagenomic analysis. Scientists are looking for changes in the viral glycoprotein spike, which mediates entry into human cells. By comparing the 2026 samples to historical Andes virus sequences from the 2018-2019 outbreak in Chup, Argentina, researchers hope to determine if the virus is becoming more adept at bypassing the human immune response. Testing is also being conducted on a respiratory pathogen panel to rule out any co-infections that might have exacerbated the respiratory failure observed in the deceased passengers. The CDC has noted that early diagnosis remains difficult, as the initial symptoms are often indistinguishable from influenza or common gastrointestinal illnesses. It is typically only after 72 hours, when the cardiopulmonary phase begins, that the specific markers of hantavirus become detectable via PCR. For the passengers currently under monitoring, this delay in diagnostic clarity represents a significant hurdle for clinical management. While the genomic investigation continues, public health guidance remains grounded in established protocols for zoonotic and respiratory protection. For the general public, the risk of a broad hantavirus outbreak remains extremely low, as the virus does not spread with the efficiency of seasonal respiratory pathogens like influenza or SARS-CoV-2. Prevention is primarily focused on the management of rodent populations and the avoidance of environments where rodent excreta may be present. In endemic areas, the CDC and PAO advise individuals to avoid stirring up dust in enclosed spaces like cabins, sheds, or storage units. If cleaning is necessary, the use of a 10% bleach solution to disinfect surfaces and the wearing of high-grade respirators, such as N95 masks, are the standard recommendations. These measures are designed to prevent the inhalation of aerosolized viral particles, which remains the most common route of infection. In clinical settings, the management of the Andes virus requires intensive supportive care, as there are currently no licensed vaccines or targeted antivirals for hantavirus. For the patients from the MV Hondias who develop severe respiratory symptoms, survival often depends on early referral to facilities equipped with extracorporeal membrane oxygenation, or ECMO. This technology essentially takes over the function of the heart and lungs, allowing the patient's body time to clear the virus and repair the vascular damage. Data suggests that when ECMO is initiated early in the course of hantavirus pulmonary syndrome, survival rates can improve to approximately 80%. However, for travelers in remote locations or on international voyages, the delay in reaching such specialized care remains a critical factor in the high case fatality rates observed. The limits of our current knowledge regarding hantavirus mutations are a central theme of this ongoing reporting. While the term designed may be used in speculative circles, the scientific community is focused on functional mutations, those that occur naturally but change the behavior of the virus. We do not yet know if the Andes virus is evolving toward more efficient human transmission, or if the cruise ship environment simply provided the unique conditions of proximity and duration required for a rare spillover to amplify. Agencies like the Pan American Health Organization are continuing to strengthen regional surveillance, including a scheduled training workshop in Panama for June 2026 to harmonize laboratory protocols across the Americas. As sequencing data from the current cluster is finalized and peer-reviewed, the global health community will have a clearer picture of whether the Andes virus represents a shifting threat or a persistent, well-understood challenge of the modern travel era. For more updates on global health surveillance and emerging viral threats, consider subscribing to this channel. The current scientific investigation into this hantavirus variant is centered on high containment laboratories, specifically those rated at biosafety level 4, which are required for handling live, infectious samples of pathogens caused that viral hemorrhagic fevers or severe respiratory syndromes. In these controlled environments, virologists are focusing their analysis on the three-segmented, single-stranded RNA genome of the virus, which consists of the large, medium, and small segments. The mutation referenced in recent reports is primarily being sought within the medium segment, which encodes the G1 and G2 surface glycoproteins. These proteins are responsible for viral attachment to host cells and the fusion of the viral envelope with the cellular membrane. Any structural alteration in these glycoproteins can significantly impact the virus's ability to bind to human beta-3 integrins, which are the primary receptors the virus uses to invade the endothelial cells lining the blood vessels. Genomic sequencing is being utilized to determine if a specific amino acid substitution has occurred that increases the affinity of the virus for these receptors, potentially accelerating the onset of the cardiopulmonary phase of the infection. The mechanism of what is being described as a respiratory shutdown is a complex immunological cascade known as hantavirus pulmonary syndrome, or HPS. Laboratory observations indicate that once the virus successfully infiltrates the pulmonary capillary endothelium, it does not typically cause direct cell death or lysis. Instead, the presence of the viral antigen triggers an intense and localized immune response. This leads to the recruitment of T cells and the release of high concentrations of cytokines, particularly in the lung tissue. This cytokine storm causes a dramatic increase in vascular permeability. Effectively, the tight junctions between the endothelial cells fail, allowing plasma to leak into the alveolar spaces of the lungs. From a clinical perspective, this results in non-cardiogenic pulmonary edema. Patients experience a rapid transition from initial febrile symptoms, such as myalgia and lethargy, to acute respiratory distress. As the lungs fill with fluid, oxygen saturation levels drop precipitously, and the cardiovascular system begins to fail under the strain, leading to the hypotension and shock that characterize the most critical stage of the illness. National and international health agencies, including the Centers for Disease Control and Prevention and the World Health Organization's Global Outbreak Alert and Response Network, are currently coordinating the sharing of these genomic sequences. The focus for these agencies is to distinguish between natural antigenic drift, the gradual accumulation of mutations, and more significant antigenic shift or reassortment. Because hantaviruses have a segmented genome, there is a theoretical risk that if two different strains infect the same host cell, they could swap segments, potentially creating a hybrid virus with novel characteristics. Public health labs are monitoring for any evidence of such reassortment, particularly in regions where different hantavirus strains, such as the Andes virus and the Laguna Negra virus, are known to overlap. Diagnostic testing currently relies on real-time reverse transcription polymerase chain reaction, or RT-PCR, to detect viral RNA in blood or tissue samples during the early stages of the disease, while enzyme-linked immunosorbent assays, known as ELISA, are used to detect IgM and IgG antibodies as the infection progresses, contrasting the more sensationalist claims found in some reports. Officials emphasize that there is currently no verified evidence to suggest that these mutations are designed or the result of anything other than natural selective pressures. Viruses naturally evolve to better adapt to their hosts or to overcome environmental barriers. The scientific priority remains the verification of the transmission route. While the vast majority of hantaviruses are transmitted via the inhalation of aerosolized droppings from infected rodents, Andes virus lineage has demonstrated a unique, albeit limited, capacity for human-to-human transmission through close contact. Current laboratory testing is aimed at determining if the observed mutations enhance the stability of the virus in saliva or respiratory droplets, which would have significant implications for hospital isolation protocols and the management of close contacts. At this stage, data remains preliminary, and health organizations are urging a focus on verified clinical outcomes rather than speculative genomic modeling. The emergence of highly virulent hantavirus strains necessitates a robust and multifaceted public health response centered on the principles of the One Health approach, which recognizes the interconnectedness of human, animal, and environmental health. Surveillance remains the primary line of defense. International bodies such as the World Health Organization and the Centers for Disease Control and Prevention maintain specialized branches, including the Viral Special Pathogens Branch, to monitor zoonotic spillovers. This surveillance involves not only clinical monitoring of human clusters, but also active field biology. Researchers trap and test rodent populations, specifically the sigmodontine rodents that serve as the primary reservoirs for New World hantaviruses, to track the prevalence of the virus in the wild. By mapping these hot zones, officials can issue targeted warnings to agricultural workers, hikers, and residents of rural areas before a human outbreak occurs. The genomic sequencing described in recent reports serves as a critical tool in this effort, allowing scientists to identify if a virus is trending toward increased stability or human-to-human potential. Though it is important to distinguish between natural evolutionary selection and speculative claims of artificial design, practical prevention strategies are rooted in the physical behavior of the virus itself. Because hantaviruses are enveloped in a lipid layer, they are highly susceptible to common disinfectants, yet they are extremely dangerous when disturbed. Public health guidance is explicit. Individuals should never use dry sweeping or vacuuming in areas where rodent activity is suspected, as these actions aerosolize viral particles contained in dried urine and droppings. Instead, the standard protocol involves wet cleaning. This requires saturating contaminated areas with a disinfectant solution, such as 1 part bleach to 9 parts water, for at least 5 minutes before wiping with a damp cloth. For those working in high-risk environments, including cabins, sheds, or crawl spaces that have been closed for the season, the use of rubber gloves and N95 respirators is considered essential. These measures are designed to prevent the inhalation of the virus, which is the most common route of transmission into the deep alveolar tissues where the most severe damage occurs. Clinical management of hantavirus remains a significant challenge due to the lack of specific antiviral therapies or a widely available vaccine. While ribavirin has been used in some clinical trials, its efficacy against New World hantaviruses, like the Andes and Sin Nombre strains, has not been definitively proven. Consequently, medical intervention is almost entirely supportive. This includes the use of mechanical ventilation and, in the most severe cases, extracorporeal membrane oxygenation, or ECMO. This advanced technology serves as a bridge, performing the gas exchange for the patient while their lungs are filled with fluid. However, the ethical and logistical reality is that ECMO is often only available in high-resource urban medical centers. This creates a critical disparity in outcomes for patients in remote or developing regions where the virus is often endemic. The international medical community continues to emphasize that early detection, noticing the rapid progression from fever to shortness of breath, is the single most important factor in surviving an infection as we move forward. The limits of our knowledge must be acknowledged with transparency and professional caution. While the identification of specific genomic mutations provides a roadmap for potential risks, it does not offer a timeline or a guarantee of a widespread pandemic. The science of virology is one of probabilities, not certainties. Public health officials are tasked with the difficult balance of alerting the public to emerging threats without inciting unnecessary panic or stigmatizing specific regions or populations.